double check. So on question number 14 of the practice EOC, you'll notice that it has cell structures, right? And it has cell wall. It has DNA. It has nucleus. It has ribosome. And then it has two columns. It has present in animal cells, and it has present in bacteria. Now, what happens with a lot of kids is they see this word bacteria and they don't remember that means prokaryote, right?
Which is non-nucleus.
That's right. And those people that do remember that that's a prokaryote and therefore no nucleus, they think that means no DNA.
No, it has
that's
DNA,
a mistake.
But the nucleus.
That's right. So when they ask you to check the structures that are present in that particular organism, then you definitely would say check on DNA. Bacteria have cell walls.
No nucleus.
Cell walls don't, so I would leave those blank. Right? So I would not check those. I would check that one. I would check that one. These definitely have DNA.
And a nucleus.
And they have a nucleus, and they have ribosome. Exactly. But these don't have a nucleus, but they do have ribosomes. They're non-membrane bonds. So these are the structures. Now, what other structures could they give you? They could give you chloroplasts, mitochondria, cell membrane, exactly. So they can give you all those organelles, and they could ask you, instead of asking you bacteria, they might ask you plants, or they could ask you fungus, right? And then they could give you these different check boxes, and they can ask you which one of these would have it and which don't. So you really just need to know what cell structures are where and what they do. If you know that, you're going to get some questions right on this exam, okay? If you don't, you can see it's pretty easy as long as you know what your cell structures are. Now, in this one, they give you a gel electrophoresis. You guys went over this in Miss Bannicourt's class, right? Cell comparison.
I don't think so.
You should have. I believe you should have. But what it is you have these little wells in a gel, and it literally looks like it's an agarose gel, but it looks like gelatin, clear gelatin. And you put some protein, and it's in a liquid, and you put some protein in here to make the protein blue. It's not blue, it's black, but whatever. And then you run electricity through the whole thing. You put a charge on one side and a charge on the other. The electricity runs in that direction, right? Electricity moves, positive, negative, or negative to positive. I should redraw that, I guess. It runs, right? And then, because the proteins are charged, they're going to move through the gel, and they're going to make these bands. And now you've seen these bands because I've showed you these bands in class, but maybe you didn't know the word electrophoresis. And the different proteins, depending on how you cut them up, but depending on the process, these could be protein, these could be DNA, they could be a lot of things. It's really not necessary that you understand that for this example, but that's what electrophoresis is. I just wanted you to know that. That's why they call it electrode. And this is electricity, right? So they're just separating the different components of the molecules by size. So the bigger ones are here, and the smaller ones are here. and they're separated by what we call molecular weight. So the bigger they are, the slower they move through the gel. Is that okay?
Mm-hmm.
You're good with that? All right, so that's electrophoresis. Now you got several cells, they say, and they go 1, 2, 3, and 4, very easy. Then they have tissue source. Now again, the molecule they can put in this machine can be DNA, it could be RNA, it could be anything. So you have to pay attention to the problem, okay? Just because they tell you DNA here doesn't mean there's going to be DNA in the next one they give you on Tuesday. All right, so it could be any molecule. Just pay attention to what they're telling you. This is an individual A and individual B, right? So there's two different individuals. Do you understand that?
Yes.
Okay. So this is the source of the tissue from person A, or individual A, and the source of tissue is individual B. It says a table shows a sample of cells taken from tissues of two individuals of the same species. DNA from each cell is analyzed using gel electrophoresis. So they're taking. So this is a cell type. Maybe this is liver, and maybe this is a liver from A, and this is liver from B, and then they put in here skin, and they do this for all of them, right? And they look and see what kind of DNA pattern you get, okay? And that pattern's going to tell you, is the DNA similar or not, right? Like, if you looked at the comparison, you'd say, like, these two are very similar, right? And this one's not similar, so maybe B is a sample of skin, right? That makes sense because the patterns match.
Yeah.
All right, so that's all that they're talking about. But you don't really need to know that for the problem. They say liver here, liver here. They say lung here, and skin here. They say muscle here, and they say muscle here. And they say blood here, and pancreas here. So different tissues. So two livers from two different people, lung and skin from different people, muscle and muscle from different people, and blood and pancreas from different people. Okay. Which prediction will be supported by the results of the gel electrophoresis analysis? So A says cell 1 so that's this one, right? Cell 1 from individual A will have an identical banding pattern compared to cell 1 from individual B. Cell 1, and so 1 will have the same pattern as B. So the question here that you have to answer is, here's the question you have to ask yourself: what am I comparing? When I say that the banding pattern for this banding pattern is the same as this banding pattern, what am I putting in here? What molecule am I putting in there? Look at the problem. Read the first sentence. What am I comparing?
The top first sentence, the question?
Yeah.
It says the table shows two sample cells taken from the tissue of two individuals of the same species. Like DNA from each cell analyzed.
DNA. So I'm comparing DNA.
Mm-hmm.
Where's the DNA going to be the same? This is something I repeated like 8 times, and in these practice questions, they ask you the question like 3 or 4 different ways. So where is the DNA going to be the same? Is the DNA ever going to be the same between person A and person B?
No, because two different.
Unless they're twins,
yeah.
Right? They're not. It's not going to be. They don't tell you it's twins, so you have to assume they're not. So these are all going to have what? These are all going to have what?
DNA?
The same DNA.
Because
even though they're different, exactly. Even though they're different structures, the same person.
Mm-hmm.
So they have the same DNA, so they're going to have the same pattern, right?
Mm-hmm.
Does that make sense? So you really didn't have to understand what electrophoresis was, but it would have helped, but it's not necessary, right? The key is what? That that everything that is from person A is going to have the same DNA, and everything that's from person B is going to have the same DNA. It doesn't matter if it comes from a pancreas or a muscle or skin. Now, I've said that many times, right?
Mm-hmm.
So these are going to have the same DNA. So what's the only answer in question 15 that gives you that answer of the same DNA?
Oh, because cell 1 from individual A will have identical binding compared to cell 2 from individual A.
So individual
it
A,
was saying that these two have the same DNA.
these two have the same DNA. Why?
Which is true.
Which is true, because even though they're liver and lung. Now, anybody, almost anybody else that doesn't understand that is going to say liver and liver will have the same, and
Yeah, but
muscle and
they're from
muscle,
different
but they're from
people.
different people that have different DNA.
Right, that's what I was thinking.
So even though the structures are different, and so that's the key. The key is that they're at what molecule are you comparing? If it's DNA, then they're the same, right? If you're looking for RNA, then the RNA in a liver is going to be different
From
RNA in a skin, because it's what's expressed, right? So the DNA, the genes in the liver are going to be more similar between person A and B, because they're for cell 1 because they're both liver. So it depends on the molecule that you're comparing. If you're comparing DNA, for sure, the DNA in the same individual is going to be the same. If you're comparing RNA, then the DNA in livers are going to be the same. The DNA in muscles are going to be the same. And not the DNA, I'm sorry. The RNA in livers are going to be the same. The RNA in muscle are going to be the same, because you need the same kinds of proteins in livers and muscles, and the same kinds of proteins in muscles. But you're going to need different proteins in the lung than you do in muscles, right?
Yes.
Okay, so that's 15. Tomorrow.
Before we do Kahoot.
Yeah, we might even just spend a good part of the day on this. Let's look at the next one.
Okay, yes, this is I was just...
You didn't like this one? All right, in this one, what they show you is they show you a red blood cell. Now, they could show you any cell, right? But they just show you a red blood cell because it's a blood cell. And they give you a key. Anytime you see a key, pay close attention to it, because it gives you a lot of information. They said triangles of protein. They said this thing here is a red blood cell. And they say this thing here is a sugar molecule. All right? So there's 3 of them. They say outside the cell you got this going on. And inside you got this. Looks like an A somewhere. Okay, so you got this key, and then it tells you that there's proteins, sugar, and red blood cell in the blood. And that's what it looks like, right? There's a little bit of sugar and a little bit of protein inside the red blood cell. All the outsides has sugar and protein, right? What is most likely going to occur? First of all, what do you know about protein and sugar? Is it gonna go from high to low without any help? Because out here is high, right? Mm-hmm. I think that's obvious based on the drawing. And in here it's what? Low. So we know that. Mm-hmm. Are these triangles and circles or dots, these proteins and these sugars, are they going to move from high to low? Are they going to diffuse? It's not going to move easily, and that's not going to be simple diffusion. You're right. It might be facilitated diffusion, but it won't be simple diffusion, right? Because they need help. They need help. Why do they need help? Because sugars are big, and proteins are big, and usually they're charged, all right? So water from the solution will diffuse into the red blood cell. Will water in is this hypertonic or hypotonic?
This is higher concentration on the outside, so it's hypertonic.
That's right. It's hypertonic. So in a hypertonic solution, what's going to happen to the water?
It moves out to in.
Excellent. No.
Well, it moves both ways actually.
But where's the majority of it going to move?
In.
No. It's going to move out in a hypertonic solution. So make sure you know that. Hypertonic solution, when it's more outside than inside, it's going to move. The water's going to move out. That's all there is to it. Why? Because remember, for all the other reasons that I discussed, go look at the video if you're confused. Okay. Hypertonic solution, the solutes can't move in, then water has to come out to try to balance out the concentration. That's what's going to happen. So water from the solution will diffuse into the red blood cell?
No, we just talked about it.
We just talked about it. And you're right, by the way. There is going to be a little bit of water going in because it is a dynamic process, right? So there's some goes in, but mostly it goes out. Water from the red blood cell will diffuse into the solution.
This will be the solution?
Yeah. Don't forget. Yeah, that's right. This is the solution. And this is solutes out there. Then this is the cell, right? So again, water from the red blood cell will diffuse into the solution.
Yes.
Yes. So we know it's B for sure, but it could be more, right? Protein and sugar from the solution will diffuse into the red blood cell.
No, we just talked about it.
Yeah.
It's hard and
It's too big and it's charged. Yeah, exactly. It could be positive or negative, and its charge is big, so they're not going to diffuse. Protein and sugar from the red blood cell will diffuse into the solution.
It's there in the solution.
Right, it's there in the solution, number one. Number two, if it was going to diffuse, it would go in. It wouldn't come out because it's going to go from high to low. And three, it's not going to pass through the membrane because they're charged and they're big. So for those three reasons, proteins and sugars can't move in and out easily, so they're not diffusing, not without facilitation, right? So the only possible right answer is B. Do you agree?
Yes.
Let's check our answer. So as you saw, B is the only correct answer, and we can move on. So it goes back to what I said when we talked about diffusion and osmosis. So let's review what the rules are. Rule number one, if the solute can move, it will from high to low, right? Two, if it can't, then the water will move in the opposite direction. All right? So if this was going to go from high to low, it would, from high to low. But it can't. So water's going to move from the opposite direction. Right? All right. So that's really the rules for diffusion. Now, what is it that determines if the solute can move? Well,
if it's non-polar and if it's small.
So small and non-polar. It can be maybe small and polar, depending on how small is small. Okay? But it's certainly small and non-polar is going to easily move. So what are examples of this? Oxygen gas can move easily. Carbon dioxide gas can move easily. They're non-polar. They're small. Water, even though it's polar, can move easily. It can move. It doesn't. It still needs help. So you have porins that allow it to go moving much faster in and out. But water does move in and out. That's how you get osmosis, right?
Mm-hmm.
So that's small and nonpolar, or small and slightly and polar, but small in any case. What does not? This is yes.
If it's charged and if it's big,
So if it's charged
especially if it's big, it's not gonna be able to get
or
through.
big, then no, it's not going to get through easily. So things like sodium ion need help, even though it's smaller than oxygen. Carbon dioxide has charge, so it can't move easily. Chlorine ion, potassium ion, these all need to help cross
That's
the
why they have the
membrane.
potassium pump to get help.
That's right. That's why you have the sodium potassium pump that uses ATP,
Which is extra energy needed.
there's energy needed because they're moving against the concentration gradient, and they need help because they're ions. So okay, so that's one thing. The other thing is proteins. They're big. Proteins tend to be big, much bigger than CO2, and sugars also tend to be big. And not only are proteins and sugars big, they tend to be charged or polar. So these are big and polar, and these are small and charged. So these don't move easily. So that's why in the problem they told you protein and sugar, because they want to know, do you know that proteins and sugars don't pass through the membrane? I mean, if you don't know that, you can't answer this question. It's a guess. And that's a bad guess because they made the answers very close to each other. All right, so you have to know these. This is why I grilled you on this stuff earlier in the year.